Dr. Jim Hood. Both will be talking about watershed sciences and water quality, particularly in the Lake Erie watershed. So we are very excited about having them here and look forward to hearing about their fantastic research. Dr. Margaret Kalcic will speak first. She's an assistant professor in the Department of Food, Agriculture, and Biological Engineering here at OSU, and she's also a core faculty at TDAI. She has a bachelor's degree from Olin College of Engineering, master's and PhD from Purdue, and then she did a postdoc at the University of Michigan. Her research areas are in watershed hydrology and watershed modeling. She has a broad and applied research program which spans many different fields, including water quality modeling, but also geospatial analysis and stakeholder engagement. She has recently worked on watershed modeling in the Western Lake Erie, which is what we will hear about today. And she hopes that this work will engage policymakers to incentivize and adopt effective conservation strategies to tackle some of the problems we have with Lake Erie's nutrient inflow. So without further ado, I'll let Dr. Kalcic take it over.
Thank you so much, Marta. All right. Well, thank you for inviting me to speak in the TDAI 2021 speaker series. Today, I will share with you some of the latest research that we are conducting on watershed modeling in the Western Lake Erie watersheds, specifically to support water quality management. The overall goal of my research and outreach program is to increase adoption of effective agricultural practices or agricultural conservation for the purpose of protecting water quality and the environment. This is a list of some agricultural practices that people might use to do such a thing for those that don't have an agricultural background. I have learned from my colleagues in the social sciences, especially, that a lack of clear science about conservation effectiveness, do these practices work, is a barrier to land managers adopting conservation measures. So scientists like myself, and perhaps Jim is on the fringe of this as well, we need to measure the performance of conservation practices in the farm fields and streams around them, and measure or model the water quality benefits at the watershed scale. So the science can then be clearly communicated to stakeholders, policies, and land managers. And to do this well, I find the need to cross disciplines, particularly with social scientists. In today's talk, I will focus mainly on recent modeling work that we're doing to inform agricultural management in Western Lake Erie watersheds, but there's a little more that I'm not showing you that we could discuss offline if you're interested. Lake Erie has a long history of impairment. The western basin of the lake has been plagued by algae blooms before, and it is once again. Nutrients, and particularly phosphorus, drive algal growth in the western basin's shallow, warm waters. Phosphorus comes from point sources, such as water treatment plants, or wastewater treatment plants, as well as non-point sources such as farm fertilizers. Historically, detergents were also a major source of phosphorus, and today agricultural sources are dominant. You may know all of this already, especially those that have spent a long time in Ohio. The watersheds draining to the western basin of Lake Erie are heavily agricultural. The largest watershed in the entire Great Lakes region is the Maumee River watershed. And today, it's incredibly productive row crop agriculture, but once, formerly, it was mostly the Great Black Swamp. So we've got a lot of low topography, poorly drained soils, and very productive agriculture once we've drained those soils. In 1978, the US, sorry for these animations, the US and Canada set a phosphorus target or maximum at 11,000 metric tons of phosphorus per year. Point sources worked hard to reduce their phosphorus loading, and these targets have generally been met thereafter. Yet in the early 2000s, algae blooms of increasing size and toxicity have become commonplace. In 2016, new targets were developed using an ensemble of lake models to better understand what we really need to do to clean up the lake. These new targets are more nuanced. Instead of an annual maximum for the whole lake, they require a 40% reduction of the 2008 values, which hit close to the old target, as you can see on the graphic there, for both total phosphorus and the dissolved reactive form of phosphorus, also called orthophosphate or soluble reactive phosphorus. A lot of names for that, and I might use a few in this presentation, as well as annual and March through July time periods. Additionally, in recognition that a particularly wet year might cause an exceedance every once in a while, these new targets are to be met 9 out of 10 years. Finally, the new load targets are accompanied by concentration targets. So these are much more nuanced targets than we had before. But we still have even if we know maybe what the solution needs to be in water quality terms, we need to know what can get us to that solution in the landscape. How do we achieve these targets? A 40% reduction of phosphorus from agricultural lands will require significant changes to farming practices upstream. Given the suite of known practices, we have some guidance from the science about practices that control soil erosion and surface runoff, and perhaps others that control dissolved nutrients and subsurface drainage. These practices generally come with some cost to the producer, and this is just a schematic. It's not actually, these aren't in the right places, okay? But if we apply the appropriate practices across large swaths of farmland and allow producers at the same time to make decisions on their own land, it's a great challenge to get to a solution. Add to that the multi-state and binational collaboration that's needed to manage this lake. We're lucky that here in Ohio and the other states and province draining to the lake, we have made considerable strides towards a solution policy-wise. As a researcher, I find that Lake Erie is a great place to be doing our modeling work. There are great needs, and those needs drive collection of data. They mean that we build partnerships between researchers and practitioners within and across multiple states, and we have greater support for our research. This map shows, and Jim, I stole this from you, I think. This map shows many of the monitoring gauges and collection of water quality data from large rivers to small drainage ditches or even the edge of field. And it's an exciting time to be working in this space. Hopefully the work we're doing here will also help managers elsewhere to make conservation solutions that work for their states or their watersheds. Given the scale of the problem, Lake Erie, the nature of the source of the problem, these large swaths of agricultural land that are difficult to monitor, to measure the impacts we're making, and the need to predict the impacts of changing land use and climate, because those also are going to affect our solution, we need to simulate these large lands and watersheds to plan and evaluate our plan adaptively over time. Watershed models are one tool that we can use to simulate the impact of conservation actions on flows and nutrient loads in watersheds. So the SWAT model, or soil and water assessment tool, is what I'm talking about that I use in my research. It's popular for use in agriculturally dominated watersheds worldwide and has been used extensively in Western Lake Erie watersheds for many years. The model takes inputs of topography, soils, land use, climate. It runs at a daily time step, and it simulates water and nutrient movement, soil erosion, and crop growth. To look at the spatial delineation or resolution of the model, let's consider our latest version of the Maumee River watershed model. The model breaks up the watershed into sub-watersheds, also called subbasins, and hydrologic response units, or HRUs. In this model, we have set up these smallest land units to approximate farm fields as shown in yellow for the ease of inputting known management, crop management operations, and field characteristics. The blue lines show the locations of rivers in the model, and SWAT has algorithms to calculate and route the flow, nutrients, and sediments downstream and to the lake. This model was recently published in my student Anna's paper, where she investigated accuracy of the upland watershed processes using this model, along with 2 other versions of the model developed by our team. Now let's look at the vertical units of the model. Each spatial unit has soil layers and characteristics taken from the Surgo soils database. We have specified depths to impervious layer and tile drains. These subsurface drains are, for those that don't know, they're pipes underneath farmland about 3 feet down that drain the water table down to that level so that we can, so that you can farm that land. As well as land cover and crop rotations are in the model. And the top centimeter of the first soil horizon is susceptible to sediment and nutrient transport and surface runoff. And the upper soil layers are important for plant growth and subsurface drainage. So hopefully that gives you a broad overview of what the model is to see how we can use it to simulate these nutrients and flows within a watershed. Let's dive now into some of the recent modeling work that we've conducted to support decision makers in these Western Lake Erie watersheds. I will focus the content of this presentation on a unique collaboration of modelers in this area, though there's a lot of other work that I'm not sharing with you that I'd be excited to tell you about too. Because so many separate teams have developed models in this same exact watershed, we were able to pull together a team of 6 research groups, 5 using the SWAT model, and Dale Robertson at the USGS using the Sparrow model. Starting in 2015, we've been working together to provide guidance for managers. In the largest project, we had 3 goals. First, and most responsive to the stakeholder immediate needs, we used the multiple models as a way to show paths to reach Lake Erie's nutrient targets and to show a range of expectation around the mean or the central tendency, similar to how climate models are used in an ensemble to predict future climate conditions. Secondly, we found that stakeholders were often asking questions about our watershed maps, like we have these spatial units, can we use them somehow? And they want to know what are the hotspots or which land is most responsible for this problem, also called critical source areas, that they could consider in targeting their efforts. Having multiple models of the same location gave us that opportunity to assess the uncertainty that we have that's associated with those predictions, which we suspected would be considerable, so that we could give managers not only, well, here's what the maps are, but here's how uncertain we are about those results. Third, we ran an ensemble of climate models with an ensemble of these SWAT watershed models to investigate the effect of future climate on hydrology and water quality. This work was funded primarily through the Ohio Department of Higher Education. One of my favorite parts of this collaboration was the input and collaboration that we had with a stakeholder advisory group. This group is changing over time, but this is a snapshot of many of the organizations that have participated actively in advising our modeling efforts. You can see it's a diverse group with perspectives from agribusiness to environmental advocacy, on-the-ground conservation planning to policymaking. It is through this group that I have come to appreciate the diverse needs of land managers, and this group has also birthed and honed some of the most fundable and interesting research questions that we've been working on since this project. In short, we're indebted to this group, and I can highly recommend involving stakeholders directly in research projects, provided that you have the resources to maintain those relationships, because it takes time and energy, the right people around the table, so you're getting a good diverse perspective, and that the project is meeting stakeholder needs so they have a reason to be at that table. For the first goal of simulating a path to reach new water quality targets, we conducted a sensitivity approach by altering individual practices across the entire watershed. That was the first thing that we did. We compared the total phosphorus and dissolved phosphorus concentration changes for each scenario. So this is at the watershed outlet, how much of a benefit are we getting in our water quality? The box plots show the range of the 5 SWAT models prediction over 10 years. We found that the models agreed about certain practices, such as broadcast, putting your phosphorus fertilizers directly on the soil surface increases phosphorus concentrations in your surface runoff. Others were uncertain. One of the most effective practices was subsurface applied phosphorus fertilizer, so getting it into the soil or under the soil. And the nutrient management scenario that included this was also quite effective, which I've circled there. In some cases, practices such as cover crops or wetlands were quite effective for total phosphorus reduction, keeping that sort of sediment bound phosphorus, holding that back from continuing downstream, but not so effective for dissolved phosphorus in the model. After discussing single practice scenarios with the stakeholders, we bundled or combined these practices at rates that were considered by the stakeholders to be more feasible for the watershed to achieve, though we did go to pretty high rates because we knew it would be a heavy lift to achieve our nutrient targets. So this graph shows the number of years meeting the target out of 10. Remember that nuance to our targets. And the green band is the required 9 out of 10 years. We want to hit that green band at the top. The box and whiskers are from the 5 SWAT models again, an average over 10 years of climate data from 2005 to 2014. And we found that for total phosphorus, it was possible to reach the target load 7 to 9 years out of 10, depending on the suite of practices used, as well as whether they're targeted to the highest emitting phosphorus emitting fields in the model or placed at random. The situation was more challenging for dissolved reactive phosphorus, with models only meeting the targets 4 years out of 10, only 1 more year than in the baseline model, despite all that added conservation. You can see those rates of those different conservation practices, but it's basically, 3 new practices on half of farmland, something like that. It's a lot of practices to not have much benefit for dissolved phosphorus. Therefore, realistic scenarios were promising for total phosphorus, but revealed concerns about dissolved phosphorus being more difficult to manage. This work was recently published in the Journal of Environmental Management with Jay Martin, my co-lead on the project, leading the manuscript. All right, the second initiative taken on by the multi-modeling group was that comparison of critical source areas for flow, nutrient, and sediment export in the watershed. 5 models were used for this effort, 4 SWOT models and the 1 Sparrow model that I mentioned earlier. I'm showing you here agreement among the models in critical source areas for dissolved and total phosphorus, as well as suspended sediment, that's TSS. Each model has a different spatial discretization or resolution, so results were compiled according to these HUC-12 watershed units that you can see there in the black outlines. This is only showing the non-point source contributions, i.e. agricultural contributions, and it's back calculated to be delivery to the lake for each subbasin from the SWOT models. A critical source area was defined as the top 20% emitters. So if we had perfect agreement among the models, we would see 1/5 of the HUC-12s would be dark blue in the map. But note that for DRP, the one on the left here, the Sparrow model doesn't simulate it, so we would want to see 4 out of 5 models agreeing there. Okay, in short, we found considerable variation in critical source area prediction among the models, even though 4 of the 5 models were all developed using SWOT and had similar inputs. Sediments were predicted with greater certainty than total phosphorus, and that greater certainty than dissolved. And there are many reasons for this uncertainty, including a fairly homogeneous watershed where at the HUC-12 resolution, maybe there aren't clear hot spots and cool spots. But this, I think, would be helpful for managers to be able to know not only where we predict might be hot spots or cool, but how certain we are that those are. So some of these locations are very likely to be hot spots, and some we really don't know very much about. The third initiative I'll move on to, just note that that is published as well by my postdoc. The third initiative was to predict the influence of climate change on the hydrology and water quality in the Maumee River watershed. A handful of climate change and modeling studies have already been done in this watershed by single modeling groups, and results have varied widely. So this collaboration allowed us to assess the ensemble of models and understand the sources of uncertainty in climate change modeling. We selected 6 climate models, already downscaled and everything, for the RCP 8.5 scenario to use with the 5 SWOT ensemble. The ensembles of climate and watershed models did not find a clear direction of change for surface runoff, tile drainage, or water quality. This was a disappointment at first. We wanted to say what was going to happen in the future, and what we could say was we don't know. So how can we create clear messages for our stakeholders when models show such wide-ranging prediction? This challenge, I guess, was an opportunity because we used this lack of agreement to study the variation among the models, to study that uncertainty. So my student, Haley Kuyava, analyzed the 30 ensemble members and found that variation in predictions for discharge and nitrogen, shown in these plots on the left, were explained by the variation in the climate model ensemble. However, variation in predictions about whether phosphorus would increase or decrease in a future climate was explained mainly by the SWOT model. This means that as watershed modelers, if we want to drill down to get a clearer message of how climate change will impact phosphorus, we may be able to achieve this by improving our SWOT models, looking within those watershed models. It is important to note that the phosphorus transport has 2 primary pathways from the field to the river, from cropland to rivers. One is through surface runoff and the other is subsurface drainage. So if we, for example, validate our models at more monitoring locations, including the edge of field, we might be able to improve the pathways of phosphorus transport and thereby raise our confidence in future predictions. Haley published this work, and a follow-on manuscript is nearing submission that layers in conservation practices to look at resilience of conservation in a changing climate. This work and our stakeholder group have led directly to several more research projects. Just going to show a couple of them real quick here. We have begun another multi-SWAT project to simulate the impact of fertilizer timing based on use of this runoff risk advisory forecast that's been developed for several states through NOAA. And since running scenarios, for the second bullet here, the state of Ohio has developed the H2 Ohio initiative, that's attempting to fund conservation at much higher rates in Northwest Ohio and across the state. And we're in discussion with the state about potentially evaluating their program to provide estimation of water quality benefits required for adaptive management of that program. The stakeholders also drove our research into two new areas for me. The first is a focus on soil health and water quality at the field scale. And the second is looking at in-stream processing of nutrients as they travel from farm fields to the lake. This last one leads directly into Jim Hood's presentation, which follows mine. A few years ago, Jim and I began to collaborate in this space. And one salient experience for me was sitting in a stakeholder advisory group meeting and talking with our stakeholders about limitations in our models. And one of the heads of a commodity group in Ohio raises his glass and he shakes it and he says, "You're telling me that during big flow events, the river acts like a pipe. But I say, look what happens when I shake up my glass. What happens to the tea leaves?" In other words, the resuspension of all of these materials, the sediments and nutrient-laden sediments in the streams should be contributing potentially to Lake Erie's phosphorus load. So these producers, the agribusiness, the environmental, all of these different angles have led us to these new research topics, which have been exciting. And I'm not presenting on them today. Maybe next year or the year after, we'll have more to say on those topics. But that leads a little bit into Jim's talk, but I think we're going to take a couple questions before then. So thank you so much.
Thank you very much, Margaret. Great presentation. If you have questions, please place them in the Q&A box and I will read them off. If you want to have a more detailed discussion, also let us know. We can unmute you and you're free to ask questions verbally. So someone is asking whether you can provide these published references, and I think this talk will be uploaded to YouTube, so you'll be able to, whoever is interested can go back and check on those references for sure. And
you're also welcome to email me and I could send you the papers if that's of interest.
So while we're waiting for some questions to appear, I have a question. This is quite outside to what I do and outside of my expertise, and I'm always curious how receptive the stakeholders are to the notion of uncertainty in general. Because from my very limited experience with stakeholders in general, my research is not as applied. It's not easy to convince them oftentimes that they have a hard time understanding what uncertainty is and trusting the results that show a high level of uncertainty. So what's your take on that?
That's a great question. I've experienced stakeholders wanting to see no uncertainty in any result because that would be too complicated or might make it more difficult for them to manage. And then stakeholders saying, I'm not going to believe anything unless you show me uncertainty along with that result. So I think it varies widely. But I think as stakeholders engage more in the research process, they're more likely to want to see the uncertainty or to understand how much we trust this, right? So there are differences in what the stakeholders are going for. But for instance, when I had several meetings with the agribusiness, folks, leaders in this project, and one of those meetings, I kind of was asking, like, why are you so interested in knowing all the details inside this model? And they're like, we know this is going to be used somehow in policy, and we really want to understand it and make it right as we can. And so I think that depending on the stakeholder need, right? What is that stakeholder need? What is that stakeholder interest? And it would vary. But for those that just really want to be able to say that they trust the tool, they're going to want to dive in and they're going to want to know more. And then they're going to want to see what that uncertainty looks like. But I would say that of these studies, the first one being, how do we reach the targets? The stakeholders really wanted that one. The climate change, they said they wanted, but then it was really hard for us to make a clear message around it. And that uncertainty, it was masking any real, like, are we going to have an increase in nutrients or decrease in nutrients? And that was hard, I think, for us as scientists and also for the stakeholders to know what is the value in that work. And then the watershed hotspots work, the stakeholders didn't really ask for it, but stakeholders so frequently ask for these maps that we felt as researchers like we owed it to them to show that there is uncertainty in these maps that we produce. So I think there was mixed reception of uncertainty even in our study.
Thank you.
That's a great question, Marta, and a great discussion that I would love to continue. But I want to ask a slightly different aspect of it. So a lot of the models, they are sort of domain mechanistic models that you use for simulation. So they come from good theory, good science of understanding of how the nutrients flow and how the different processes.
I'm joining up.
Sorry, how the processes flow and how the nutrients flow. But then we try to measure them with actual data, right? And so we try to find this good intersection between what's measurable in data versus what we understand through theory and science. And so how do you bring the two together? And if we have different things that we can measure through data, can we find, does it change our models and vice versa? How do the models guide what we can measure?
Definitely. How do we bring the two together? Great question. And I think there are a lot of different ways. So a simple way that data informs the modeling is that we calibrate and validate our models against data. So we check, does it work? And if it doesn't work at matching that data to some degree of accuracy, then we need to tweak our models in some way. And in the SWOT model, there are a lot of parameters that we change in the process of calibration to reach good performance in, of those values, or decent performance. But that relies on the structure of the model being correct. If we make those changes, well, the structure of the model is correct, we make those changes and we're able to fit that data. But if the structure of the model is incorrect, then we need to actually dive into the source code and change the structure. So Jim's talk, that follows on this, in-stream processing is very limited in the SWOT model, even though it's a watershed with rivers and streams and things are happening, but are they the right things? Do we have algae represented in the model properly? Do we have the sort of uptake and release of phosphorus in the fields? Like, there's all sorts of things that we could say are limitations in the model structure, and those would require us taking that measured data, understanding, well, then what are the processes supposed to look like, and maybe building some new equations from the data and then putting that in the model. So there's different levels of using that data, I think, to improve the model. The models can also speak to what data is needed. It's not just a one-way street. So if we're modeling and we say, we really are very uncertain about this aspect in our model, but we think we need something here, but we don't think this equation is necessarily right, like, well, then what should that equation be? Or what should be the processes that are included? We need someone to measure those things and tell us what are the most dominant processes that matter and how should they be related to one another. So I think ideally all modeling work should be using data and informing what further data is needed so that those who are collecting data can get more out of the data they're collecting, and it's just better for the science.
Thank you, Margaret. I think we probably should move on to Jim's talk. We are right in the middle of our time slot. So let me introduce Jim now. Dr. Hood is an assistant professor in the Department of Evolution, Ecology, and Organismal Biology He's also a core faculty in TDAI. He's been trained, his bachelor's degree is from Lawrence University. He got his master's just down the road at Miami University in Oxford, Ohio, and a PhD from University of Minnesota. And then he spent some time as a postdoc at jointly at Montana State and as a Rani's postdoctoral fellow in Iceland at the Icelandic Institute of Freshwater and Marine Fisheries. Dr. Hood is an ecosystem ecologist who studies how human-induced stressors like climate change or nutrient pollution influence aquatic organisms and ecosystem processes. His research has focused on lakes and streams across the US, but also in Venezuela and in Iceland. And most recently, he has focused on understanding how nutrient pollution and climate change have interacted to influence algal blooms and invertebrate communities in Lake Erie. And that's what he will be talking about today. Take it away, Jim.
Thank you very much, Marta, for that nice introduction. So today I'm going to talk on some work that builds a bit on what Margaret was showing earlier. And so we're gonna ask this question about what role the rivers are playing in shaping nutrient exports, particularly phosphorus exports to Lake Erie. So you imagine your stakeholder with his water glass and he's shaking it and he's asking what's happening in the streams during high flow. That's what this talk is about. So, you know, I just want to start, I won't belabor a lot of what Margaret's already talked about, but we know that, you know, cyanobacteria blooms have been an increasing issue across the United States and across the globe over the last 10 or 20 years. And that these have caused a number of economic and human health and ecosystem service impacts across all of these systems. And as Margaret mentioned, this is also the case in Lake Erie. So the thing on your left, the picture on your left shows an algal bloom in Lake Erie in 2011. And you can see on the right, the Noah, Heidelberg et al. Harmful algal bloom forecast for Lake Erie going from 2002 through to 2019. And so in Lake Erie, there's really been an increase in the number of harmful algal blooms since the early 2000s. These algal blooms have had a large ecological and economic effect on the region. And so therefore, there's a lot of interest in understanding what's causing them and how to mitigate them. And as Margaret mentioned, the blooms are believed to be driven by phosphorus loading, primarily phosphorus. Nitrogen is also important, but it's phosphorus. It's the primary driver here from the Maumee Watershed, which is a watershed in northwestern Ohio that's about 90% agriculture. And because point source phosphorus additions to these rivers have been decreased, the presumption is that most of the phosphorus is coming off the landscape from agricultural fields. And some of this is overlapping with Margaret. That's okay. You know, so the guidelines that are developed for reducing cyanobacteria blooms to Lake Erie have focused on the phosphorus exports from the Maumee watershed during this key spring period, somewhere between March and June. And that's important because that's the period when most of the phosphorus that's supporting these algal blooms is delivered to the lake during these high flow events that the stakeholder was talking about. And I would just emphasize that as Margaret mentioned, there are targets of near 40% reduction in total phosphorus, which is all of the phosphorus coming downstream. And that total phosphorus can be broken up into sediment phosphorus, right? So phosphorus attached to dirt, and dissolved phosphorus. And so that's phosphorus that makes it through a 0.7 micron filter. And the dissolved phosphorus fraction, or the DRP fraction, is particularly important because it's 100% bioavailable to the cyanobacteria. If you give that DRP or dissolved phosphorus to cyanobacteria, they'll grow. And so there's a particular emphasis on reducing dissolved phosphorus loading from this watershed to Lake Erie. So this phosphorus management, as Margaret already mentioned, requires a really good understanding of where the phosphorus sources are from this watershed, where the sinks might be in the landscape, if there are wetlands or other sites that are retaining phosphorus, and any transformations, particularly between sediment or dissolved phosphorus, that might increase or decrease the bioavailability within this watershed. And to estimate these sources of and sinks, the framework kind of looks like this. This is a gross oversimplification of Margaret's work, and she's probably banging her head right now, but we'll enjoy that. So, you know, the approach is you monitor water quality, dissolved phosphorus, stream discharge at this downstream site in Maumee. And so here's the record of total phosphorus loading into Lake Erie from 1975 to present. And if you look within any given year, you know, the load during this key May to June period might be something like 1,000 tons of total phosphorus coming out. And since we think that point sources are pretty low, and there's this assumption that rivers are essentially pipes, that means that there's about 1,000 tons of phosphorus coming off the landscape, coming off of these agricultural fields primarily, and ending up in the lake. The same can be said for the sediment particles, the dirt, and also the dissolved reactive phosphorus, which is the most bioavailable fraction. So this conceptual model here, where stuff is just, these nutrients are just piped downstream, how consistent is that with our ecological understanding of the way rivers and streams and ditches operate? And if you ask a stream ecologist, they would say it's probably not a very good assumption. It's well, and to sort of explore that, I'm going to split this May, June, spring period into two categories. You have the low flow category, which is about 23% of spring phosphorus exports, and the high flow category, which is about 77% of spring phosphorus exports within a given year during the last decade or a decade and a half. During the low flow period, it's really well established that rivers and ditches and streams can be phosphorus sources or sinks, and they can absorb in some case, they can take up in some cases nearly 100% of the phosphorus that's coming into them. Unfortunately, within the Lake Erie watershed, we don't know very much about whether the rivers are sources or a sink and how much they are, but we know in general that that's an important phenomenon we need to understand. During the high flow period, we know that rivers can add or remove sediment through erosion of riverbanks or through deposition of sediment phosphorus onto floodplains. However, it's generally assumed that dissolved phosphorus and sediment phosphorus is just, or dissolved phosphorus is just piped downstream, like Margaret's models are assuming. And so what we're going to do today is test that assumption that during these high flow events, dissolved phosphorus, which is the most bioavailable fraction, is just being moved downstream in transport. And this is a reasonable assumption. I've made this for a lot of my life because, you know, there's a lot of water moving down during these floods really fast. So here the hypothesis we're going to test is that during spring high flow events, abiotic phosphorus cycling, I'll tell you about that in a second, either increases or decreases the bioavailability of total phosphorus loads to Lake Erie, which will somehow mediate the summer algal bloom, depending on whether it's decreasing or increasing bioavailability. And the mechanism here is phosphorus absorption or desorption. So if you imagine that this is dissolved reactive phosphorus over here, and this is a sediment particle, dissolved phosphorus can be attached or absorbed to these sediment particles. When they're absorbed to the sediment particles, then the bioavailability is fairly low. It's probably less than 20%. When they're dissolved in the water column, their bioavailability is very high, bordering on 100%. So this mechanism basically switches phosphorus from something that's very bioavailable to algae to something that's very unbioavailable and actually probably represents a long-term sink. So to evaluate this hypothesis, we're going to do a couple things really rapidly. First, we're going to just ask whether or not there's absorption or desorption in these streams during this time, during high flow events. We're going to scale this up to figure out to estimate the relevance of this phosphorus cycling to tributaries, for a couple key focal tributaries we're going to focus on here. And then we're going to scale up again to look at the relevance of this to the entire Maumee watershed and determine how it might be influencing blooms. So this work all starts with Whitney King, who was a master's student in my lab a couple of years ago and is now an EPA fellow. She measured river phosphorus cycling, so phosphorus absorption and desorption during high flow events in the 6 tributaries listed here between January and June 2019. So basically she just got in a truck every time there was a storm event with her buckets, went out, threw her buckets over the stream as is shown here, and collected some stream water, centrifuged it down, and measured absorption and desorption. 75% of her 78 total measurements were greater than 75% 5th percentile flow, which we consider high flow here. And they're all shown here on the hydrograph. So what did Whitney find? She found that in the vast majority of cases, all but one actually, suspended sediments absorbed phosphorus, right? And so that means that they're decreasing the bioavailability of phosphorus as it moves downstream. And so what this plot on your right shows is phosphorus absorption versus the concentration of suspended sediments in all the streams. And you can see that these relationships vary among streams, but in general, absorption increases with more suspended sediments, and you get more suspended sediments in these systems at higher discharges. So this also indicates that phosphorus absorption increases with discharge or the magnitude of the storm event. So the next question is, okay, she saw absorption, but do these rates matter? Is it important at the ecological scale, or can we go on ignoring it? And so our next approach is to sort of take Whitney's absorption data, leverage some ongoing water quality and hydrologic monitoring that's occurring in these watersheds. And so here we focus on just 3 watersheds where there's significant water quality data. And essentially what we do is we scale up and try to estimate what's happening as the particles that we sampled here move downstream to the site at Waterville, that's the monitoring station. We took into account a number of things that we thought might be important constraints on total phosphorus absorption, including how much phosphorus they could actually absorb and hold, the travel time and how quickly they would fill up with phosphorus, as well as interactions between sediment and dissolved reactive phosphorus. And I'm happy to talk about any of this more in the future. So the first question is that assumption, right? Are they just moving downstream so fast, there's not enough time for anything to happen? So what I want to show you first is this plot here. And so each arrow here represents the distance that a sediment particle travels, the average distance during a day, before it fills up with phosphorus and can't absorb anymore. And so what this shows you really clearly is that before, well before these sediments are exported into Lake Erie, they absorb all of the phosphorus they can, right? So they're filling up with phosphorus really quickly. The next question is, does this matter at a scale that would matter to anybody but a dorky stream ecologist? And the answer is essentially yes. When we integrate across all these high flow periods in 2019, which was a really wet year, we see that these suspended sediments from these 3 tributaries we were able to look at absorbed anywhere between 30 and 45% of the dissolved reactive phosphorus that was going downstream of the site before it hit Lake Erie. So 30 or 45% of that phosphorus is just disappearing and getting attached to sediment particles. That's a pretty big deal. So the next question we wanted to ask, that's almost a 50% reduction in the bioavailability of phosphorus loads. So the next question we wanted to ask is, what's the relevance of this at sort of the Maumee watershed scale? And so to estimate that, we leveraged this, what is this, almost a 50, it started on my birthday, or near my same year I was born. So a 45-year record of water quality monitoring here at Waterville. We leveraged Whitney's absorption data and combined that with suspended sediment concentrations to estimate phosphorus absorption for each day during a high flow event between March and June. And so from now on, everything that I'm going to talk about integrates across these high flow events between March and June as it has been integrated to the year scale using these constraints. And so what I'm showing here is the dissolved reactive phosphorus or the bioavailable P load within each year over this time series during our window. And then I'm going to add on top of this how much absorption is affecting this. And so you can see that in the absence of absorption, which is these gray dots, you have a considerably higher phosphorus concentration, but the amount of absorption kind of visualized by the pink band is varying considerably from year to year to year. And if we look at this as the relationship between phosphorus absorption over here and dissolved reactive phosphorus load over here, you see that there's quite a lot of variation. And what's nice about these plots is that the dashed lines are the percent of dissolved reactive phosphorus that's absorbed by suspended sediments. And so you can see that sediments in any given year are absorbing anywhere between 20% and 300% of dissolved reactive phosphorus loads. And that you'll also notice that after 2003, there's far less absorption than before 2003. And I'll return to that in a second. But first, I just want to focus on this period and kind of update the simplistic conceptual model that we put together. So here's our model over here, where we have pipes moving the steps downstream. These processes I'm talking about are still not influencing the total amount of phosphorus that's moving downstream. What they're impacting is where the phosphorus is partitioned. So what we're estimating is that the amount of sediment phosphorus as things move downstream is increasing, and the amount of dissolved reactive phosphorus as stuff goes downstream is actually decreasing. And so this is really important for two reasons. One, because sediment phosphorus goes out in the Lake Erie, it rapidly settles to the bottom of the lake, and because of the conditions there, it basically represents a long-term phosphorus sink. So that stuff's not contributing to blooms. Whereas this DRP, bioavailable phosphorus, or DRP, is decreasing, and that's having that's inhibiting cyanobacteria blooms. And so what we did is we took one of the models that's used in the, simplistically, one of the models that's used in the HAB forecast, and used that to estimate the effect, what cyanobacteria blooms would be in the absence of absorption. And so shown here is absorption, the max cyanobacteria index that was observed in each year between 2003 and present. And then, so you can see 11 and 15 were the big years up here. And then we also estimated it in the absence of phosphorus absorption. And so what this shows is that these river phosphorus cycles absorption is constraining blooms by around 60% on average since 2003. So they're having, these river phosphorus cycles are having a big effect on bioavailable P, and also a big effect on algal blooms. And then finally, sort of at the last part of this, I just want to get to this big difference in absorption between 2003 and before and after 2003, which is shown here. And this is going to go really fast. I don't have time to develop it, but I think the implications are really important. So I'm going to tell you ahead of time what it says. And that's basically that this difference in absorption is due to the amount of suspended sediments, which can be linked to agricultural practices focused on reducing erosion. So what we've seen in the Maumee Basin is that suspended sediment loads to the lake, once you account for changes in discharge over time, have been declining since 1975. And that's what's shown here. So what we're going to do here is we're going to account for those changes in suspended sediment loads and estimate what absorption would have been if we had, say, 1975 suspended sediment loads. And we're going to do that by looking at this relationship between DRP load and water yield or discharge. And you can see that for any given discharge after 2003, you have much more dissolved reactive phosphorus going into the lake than before 2003. And so the next plot I'm going to show you incorporates, basically says, well, what would happen if we had 1975 sediment concentrations in there? What kind of DRP concentrations will we predict? And what we get is basically no difference between before and after 2003, once we account for that decline in suspended sediment loads. And that's what's shown here in these sort of salmon points. There's no technical difference between these lines. And so what this indicates is that agricultural practices that were likely indicates is that agricultural practices focused on decreasing erosion have inadvertently led to an increase in bioavailable phosphorus exports to Lake Erie and have contributed to these blooms. And so just to kind of sum up this part, I'd like to point out that the Maumee River is really providing an unrecognized ecosystem service by decreasing the bioavailability of phosphorus loads to Lake Erie, as shown here. That it's highly likely that similar processes may be happening in other river systems across the globe, although it's not clear whether or not sediments are a source or a sink of bioavailable peat. The other point I'd like to make is that efforts to reduce blooms and understand where to target these efforts really need to take a watershed scale approach that considers interactions between what's happening on the landscape and how that's influencing patterns in rivers and wetlands, as well as considering multiple constituents like sediments and nitrogen and phosphorus. And then the final point I'd like to make is that we need to consider the impact of climate change on this. And so our next step here is a collaboration with Margaret and Sushant Mahen to update the SWAP models to incorporate stream and river phosphorus cycling. We have a collaboration between Margaret, Laura Johnson, Becky Creeling at the USGS, Nate Manning at Heidelberg, and Chris Spies to determine the spatial variation in phosphorus cycling among these tributaries across the Maumee watershed during both low and high flow, sort of addressing those earlier questions. And then finally, we have a project that's looking at these sort of hot moments of phosphorus cycling. Are there times of the year when there's a lot of phosphorus cycling? And that's really difficult to get at with labor-intensive sampling that, like Whitney was doing with the buckets. So we're working on developing models which will utilize high-frequency dissolved reactive phosphorus and other data sets to estimate phosphorus cycling through time. And with that, I'd be happy to take any questions, and thank you for listening.
Thank you, Jim. There's a question in Q&A, so I will read it out loud. From Joseph Conroy. Have you explored the potential biological processing of payloads? Sorption is important, of course, but what about algal phytoplanktonic use?
That's a really good question, Joe. The answer to that is both yes and no. I think it's yes in the sense that we've thought about it, and Whitney even did a little experiment that sort of tried to mimic the effect of absorption and look at the stuff that was sorbed was actually as unbioavailable as we thought, and that was pretty unexciting. It basically said, yeah, the stuff is like, if you absorb it onto particles, it's about as bioavailable. It becomes less bioavailable. I think your other part of your question focuses on, what might be happening in these rivers. Are there algae that take this up, and as it's moving downstream, perhaps? And that's a really good question. And the answer is that buried deep in our methods, basically, there's two ways to do these absorption measurements. One approach is just essentially take the dirt, kill everything, take a suspended sediment sample, kill everything in there, and measure it. And what we did instead was take kind of a more holistic, non-biogeochemical approach, and we took the suspended sediments and we just measured absorption under natural conditions, except for in the dark. And so that would have kept these algae and bacteria that might also be contributing to absorption alive. So we lost some resolution on the specific biogeochemical processes, but we got a more holistic perspective, which I think you're kind of pointing at here, I hope. That was a good question, Joe. Thanks for coming.
Okay. I have one more question as well. Can you speculate, or maybe there are clear projections on how precipitation and river flow will change in Ohio with climate change? I know Ohio is more susceptible to floods than it is to droughts. Do you think that will remain the case? Do we know? And how will that, if we know, how will that affect the payloads?
That's a really complicated question. And so the projection is that precipitation will increase by about 20%, 15 to 20% maybe. Margaret has some work that relates to this that she should really describe, but that seems to suggest that that may not influence phosphorus loads as much as, discharge as much as that might predict. So a 20% increase in spring precipitation might not get you a 20% increase in loads. Margaret's here, so she can correct me if I'm wrong. And then it gets really complicated from there. So it depends on how it affects erosion, and then it depends on what it does within the lake. So one of the things that it concerns me is how that influences the lake level and sort of the how stable this phosphorus sink at the bottom of the lake actually becomes if the lake level kind of routinely gets higher and higher, and the climate gets warmer.
Mm-hmm. One last question from Tanya to both of you, to Jim and Margaret. Is there a tension between understanding the processes and actionable for stakeholders and policy outcomes, especially from the model data side?
Margaret, you've been at this a lot longer than I have.
Probably there is. I think sometimes that tension is evident in the fact that some people have a bias towards one or the other. Some people really want to see measured data. Other people really want to see models. Some people don't trust models. But like, is that a tension that's across all of stakeholders and policy? I think in Ohio, and through working with our stakeholder advisory group, I think that there's a real interest in seeing both, and that policymakers are prioritizing both of these. They're thinking hard, like, is this a question we want to answer with measured data, or is it a question we want to answer with models? And they write these RFPs for the harmful algal bloom research initiative through Ohio Department of Higher Education, which is what funded both of the projects that we focused on in this presentation. And their RFPs are like, this Ohio Department of Agriculture wants someone to look at measuring this, and Ohio EPA wants someone to model that. Like, they specifically are trying to figure out exactly what they need and get there with measured data and modeling. But it has taken, it takes some time, I think, for people to build trust and to know what questions they're really asking as well. But I would say there probably is a tension, and it's something that we've had to navigate. And there are some people that just have biases against or for certain methods, and that's always going to be the case. That's not a great answer, Jim. If you have anything you want to add to that.
So much better than what I would have said. Thank you.
We have time for one more question, and there is one more question in Q&A. Is there anything special about 2003 when we see this first major algal bloom set the new trend in the Western Basin? Looking at the graphs of suspended sediments and DRP, they appear to be long-term trends toward higher bioavailability. Do you have a theory as to what caused that sharp change in 2003?
No. Part of 2003 is just to take something that's there's two answers to that. I seem to always do that. One is that 2003 is just a really nice break point in a lot of these relationships, and so it's a good way to sort of make a binary break between two things. But there are some analyses that support that say that there's a clear break in the dissolved reactive phosphorus and maybe, I think it's dissolved reactive phosphorus loading relationship right around 2003. And one thing I didn't emphasize here is that the change in this, the explanation for, this increase in DRP is really poorly understood. There's a lot of hypotheses out there, but very few good large-scale empirical estimates of why this increase has been occurring, or why there might be some difference here in 2003.
Okay, well, if there are no other questions, we probably should wrap it up anyway, since it's past 1. Please help me thank Margaret and Jim for joining us today and for giving a fantastic presentations. It was really great talks, and I learned a lot. So thank you very much. And yeah, thank you.
Thanks, everybody.
Thanks so much.
Bye, everyone.